Right, so now discussing what is functional profiling, and I'll be discussing some questions and their answers as well. So, what is functional profiling in metagenomics and why it matters? So, it's the process of converting raw sequencing reads from shotgun metagenomics or inferred from marker genes into estimates of the genes, enzymes, reactions, and metabolic pathways encoded by the microbial community, and by extension, the biochemical activities the community can carry out in a given environment. Right. So now talking about some reference databases, what they are, how they differ, and why they matter. So first we are discussing MetaPsych. So it is basically a curated compendium of experimentally elucidated metabolic pathways across all domains of life. It contains reactions, metabolites, enzymes, and gene annotations. So it basically has around these many pathways, 17,780 reactions, then metabolites, and this pathway centric and carefully curated from literature. How it is used? It is used we first map gene families to metapsych reactions and pathways to compute pathway abundances. Because metapsych is curated, pathway assignments can be high quality but may miss poorly characterized or novel variants. Then we have KEG, which is a broad hierarchical resource linking genes, orthologous groups, modules, reactions, and pathways. And we have COG, which is a cluster of orthologous groups. You don't have to get into the depth of these or the descriptions. You can just remember the full forms. Then we have CASI, that is carbohydrate active enzymes. Then ARDB, antibiotic resistance databases, Uniprot, right? And then we can move on, Diamond, etc. Then we have hidden marker models, then human N2, and mostly all this part is something that you won't have to go into the depth of, right? So we can skip this part and we'll move on to the next part, that is this. So functional profiling, two principal approaches we have. First is the read-centric approach, that is read-centric mapping. So we map raw reads directly to functional reference databases like Uniprot, KEGG, Metapsych that we discussed. Then we get the gene family counts and finally collapse to reactions or pathways. Now, when it is good for when we know the organisms, when we have adequate reference genomes, quick surveys, when we want to conduct, or when we have gene pathway quantification for comparative analysis. And then we have assembly or genome centric. Then what we do is basically assemble the reads, then predict the genes and annotate these genes, then build genome scale metabolic reconstructions, gems, or profile functions per mag. So again, this part you can skip. Right, moving on. Okay, so now talking about assembly, what, why, and the big picture. So the goal of metagenomic assembly is to convert millions of short sequencing reads into longer contiguous sequences, contigs, right, and ultimately reconstruct genomes, that is mags, metagenomes. So this enables gene context, strain resolution, novel organism discovery, and genome-based metabolic reconstructions. Now moving on. So we have a two assembly paradigm: de novo assembly and reference guided assembly. So de novo assembly uses no template or reference. It is required when environments contain genomes absent from databases. This is best for discovery. And then we have reference guided assembly, that is, it uses a close reference, useful for variant calling and polishing substrains, but biases results towards known genomes. Right. Now moving on, we have two graph models that we'll be studying. First is Hamiltonian and second is De Bruijn. So Hamiltonian, or the overlap graph, is basically where we have nodes as k-mers or reads, edges connect adjacent k-mers or reads that overlap by k-1. In theory, you want a Hamiltonian path that visits each k-mer or read once to reconstruct the genome. This formulation underlies overlap layout consensus assemblers. Then we have De Bruijn graph, which is the practical default for short reads. Nodes represent k-1 mers or overlaps, and edges represent k-mers. Traversing edges spells sequences, repeats create loops and branching. And De Bruijn graphs are memory efficient and scale for many reads, but require choosing K carefully. Okay, moving on. So now graph simplification, tips, bubbles, and strain variants. So definitions I'll discuss first. Tip is basically a short dead-end path normally caused by sequencing errors or low coverage. Is a short dead-end path normally caused by sequencing errors or low coverage. And a bubble is two alternative paths between the same start and end nodes, which can reflect true biological variation or minor strain or sequencing error. Now assembler behavior. So assemblers remove the tips and compress bubbles to simplify graphs. But in metagenomes, these structures may represent real minor variants, right? Or basically, over aggressive pruning loses rare genuine variation. Conservative pruning leaves complexity, right? So modern assemblers try to store and annotate alternative paths. Now moving on, complexity, volume, and resolution, three metagenomic challenges. So many species, many repeats, and strain microdiversity leads to higher complexity, which leads to a tangled graph or a complex graph. Then we have volume. So metagenome projects can be very large in sample size or sequencing, which causes heavy IO and memory. And then we have resolution, which is basically assembling low abundance genomes and separating closely related strains requires depth plus smart algorithms. Now, now clustering strategies we have. First is taxonomy guided. That's classify reads, Kraken 2, Kramer classifiers, and assemble per taxon cluster. Fast, but it requires good classifiers. And second, we have composition based, which is dependent on GC content, tetranucleotide, TNF, 4-mer signatures, or 4-mer K-mer profiles to cluster the reads from novel organisms. It works when taxonomy is unknown, but signature is consistent. Then we have multi-assembly and co-assembly definitions and trade-offs. So multi-assembly is basically assemble each sample independently, assemble each of the samples independently, then we merge or compare assemblies. Lower compute per job preserves sample-specific strains, less sensitive to for rare species. Co-assemblies pool reads from multiple samples and assemble together. Higher sensitivity to rare species, but much higher compute. Risk of chimeric contacts across samples and loss of sample specificity. Right. Moving on, we have some popular assemblers like MegaHit, Metaspades, and MetaVelvet. So MegaHit basically is De Bruijn based, the De Bruijn graph based, and it is designed for speed and memory efficiency. Pre-filters low frequency k-mers to remove errors, good for very large data sets, but can be less sensitive for low coverage genomes. Used when we need to scale and we have to prioritize our speed. Then we have Metaspades, builds the De Bruijn graphs and converts to assembly graph, uses strain-aware heuristics again. Moving on, MetaVelvet basically extends the velvet for metagenomes, uses graph topology and abundance coverage to separate overlapping graphs from related species. It is good historical tool, but less used than Metaspades and MegaHit now. So basically, MegaHit used for speed, a large cohort, and many samples. Then, if you want high quality recovery of diverse strains, use Metaspades. Right, moving on from context to genomes. So, objective is to group context into bins that ideally represent individual genomes, MAGs. So, common features used are tetranucleotide frequency (TNF), composition signature, context from same genome have similar TNF vectors, and read coverage, that is abundance across samples. Context from the same genome co-vary across multiple samples, powerful in multi-sample and co-assembly contexts. So we have the most famous algorithm, that is concoct. So we use a Gaussian mixture model on TNF, that is tetranucleotide frequency, and multi-sample coverage tailored for co-assemblies. It needs several samples for robust coverage covariation. And we have other examples of algorithms like MaxBin2, GroupM, where each has some pros or cons. Often recommended to run multiple binning tools and aggregates for consensus. Now, Mac quality control. So CheckMM and my Mac standards. So CheckM approach predict the genes in bins, detect lineage specific single copy marker genes, that is SCGs, single copy marker genes, and use presence or duplication patterns to estimate the completeness, that is the percentage of expected single copy marker genes observed, and contamination, that is percentage of single copy marker genes present in greater than one copy, that is more than one copy. Unexpected duplications indicate contamination or strain heterogeneity. Right. And we have MAMAG, that is minimum information about metagenome assembled genomes that report completeness, contamination, N50, number of contacts, presence of rRNA or anti-RNAs, etc. Right. Moving on, strain variation profiling. Why strain resolution matters? So strains of same species can differ in pathogenicity, antibiotic resistance, metabolic capabilities. Right. So we have two main approaches, that is SNP based, like strain-flan, constraints, less-man, etc., and gene presence based, that is pan-flan, pangenome methods, right? So strain-flan pipeline we'll be discussing in the next video.